रिकॉर्ड टू द क्लाउड कर दिया मैंने, ठीक है, और म्यूट कर। Good morning, everyone. The topic of today's presentation is genetics of anxiety disorders, and my moderator is Dr. Shobhit Garg, sir. So, as we all know, what is anxiety? Anxiety is a diffuse, unpleasant, vague sense of apprehension, usually accompanied by autonomic symptoms. So, anxiety and fear, as we all know, normal responses to a potential threat or an actual threat. And because these are adaptive and these are important for survival, so these have been evolutionarily conserved. But whenever these symptoms, they are exaggerated or they're prolonged in a manner that they're disturbing any person's life, so then they become pathological anxiety, and that is known as anxiety disorder. So nearly 1/5 of adults report a lifetime history of one of the major anxiety disorders, and 1 in 10 suffer from a current anxiety disorder. So this is the classification as given in ICD-10, ICD-11, and DSM-5. So the ones which are highlighted with color red are same in all the three. So generalized anxiety disorder, panic disorder, generalized anxiety disorder, panic disorder, specific phobia, social phobia, and agoraphobia are present in all the three classifications. Apart from that, in ICD-10, we have mixed anxiety and depressive disorder. In DSM-5, we have separation anxiety disorder, selective mutism, substance-induced anxiety disorder due to another medical condition. And in ICD-11, just as in DSM-5, we have separation anxiety disorder, selective mutism, and other specified anxiety disorder. So anxiety disorders, as like all other disorders, they are complex and they have their etiology as in genetic and environmental. So the etiology is a combination. It derives from both genetic and environmental factors. So in this presentation, we'll be going through the etiology of anxiety disorders in the following manner. First of all, we'll be reading about genetic epidemiology. Next, we'll go through molecular genetics, and next, gene and environmental interaction or epigenetics. In genetic epidemiology, you all know we'll be going through family studies, adoption studies, and twin studies. In molecular genetics, we'll be going through linkage and candidate gene association studies and genome-wide association studies. So what is heritability? So heritability, as you all know, it is the proportion of phenotypic variance which is attributable to genetic variance. That is, it is the extent to which genetic individual differences contribute to individual differences in the observed behavior. So let us now come to some definitions. So identical twins, fraternal twins, concordance and discordance. Identical twins, we all know, are monozygotic twins. So one sperm fertilizes one ovum, it becomes a zygote, and a zygote, it breaks down, it splits into two. So there is 100% same DNA in monozygotic twins, whereas in fraternal or dizygotic twins, two zygotes are formed. Two eggs are fertilized by two sperms, individual sperms, and these are genetically similar to ordinary siblings. They share 50% of the DNA. Then concordance and discordance. Concordance is the probability that a pair of twin individuals will both have a certain characteristic, given that one of the pair has a characteristic. That is, twins are concordant when both have or both lack a given trait, whereas discordance is when a similar trait is not shared by both the twins. So we have concordance rate, which is the percentage of cases in which both members of a pair have a particular attribute. And concordance, it is given as number of concordant pairs divided by number of concordant pairs plus number of discordant pairs. Odds ratio. Odds ratio is a measure of the association between an exposure and an outcome. That is, the odds ratio represents the odds that an outcome will occur given a particular exposure compared to the odds of the outcome occurring in the absence. So odds ratio equals to 1. That means exposure does not affect odds of the outcome. Odds ratio greater than 1 is exposure is associated with the outcome, higher odds of the outcome, and odds ratio less than 1 is exposure associated with lower odds of the outcome. So now coming on to the etiology after the definitions. So first of all, we'll go to genetic epidemiology. What is genetic epidemiology? It seeks to understand how diseases and how their risk factors they are distributed in the families. So in genetic epidemiology, we'll be studying about family studies, twin studies, and adoption studies. Adoption studies are not available for anxiety disorders. So we'll be focusing on family studies and twin studies. So what are family studies? In family studies, we compare the rate of the illness in relatives of the case probants, that is those who are affected. The first degree relatives of the patients, they are compared to the first degree relatives of healthy control. So higher rates, case proband, anyone who is affected by the disorder. The first degree relatives of an affected person are compared with the first degree relatives of a healthy person. So the higher rates in the former group of the relatives as parameterized by relative risk or odds ratio greater than 1 suggest familial aggregation. Whereas now twin studies, they compare the resemblance of a condition between both the members of a twin pair using the fact that identical twins, as I said, share 100% of their genetic sequence, whereas non-identical or dizygotic twins share on an average only 50% of the DNA. Now, a 2001 meta-analysis which was done, it summarized the following findings. So the results suggested an overall moderate level of familial aggregation and odds ratio of 4 to 6. That means the first degree relatives of someone who is affected by an anxiety disorder are 4 to 6 times more likely to develop the disease in comparison to a random individual, and a heritability of 30 to 50%. That means the genetic and non-genetic factors are equally responsible for the etiology of anxiety disorders. Now let us come to, we'll be studying about 3 disorders here which have been studied: panic disorder, generalized anxiety, and phobia. Other anxiety disorders have not been much studied, and in amongst these 3 also, panic disorder is the one which has been majorly studied. So in panic disorder, when family studies were done, so data from 19 controlled family studies overall support the familial aggregation of panic disorders with risk ranging to 3 to 17 in first degree relatives. That means the first degree relatives are at a higher risk of 3 to 7 times higher, 17 times higher risk than a healthy person of developing the disease. So regarding twin studies, 2 major studies were done. First one was Watsput and second one was VET. So first one was Virginia Adult Twin Study of Psychiatric and Substance Use Disorders, and another one was Vietnam Era Twin Registry. In the first study, 9,000 twins were taken. In the first study, 9,000 twins were taken from both the sexes as well as opposite sexes. Whereas in VET, a comparable size was taken, but only US males were taken. Male twins who served in the Vietnam War were taken, and both the samples had reported that higher monozygotic than dizygotic concordance was there. Now, in generalized anxiety disorder, as we talked about, the odds ratio has been seen, it comes to be 6.08. That means they are at more risk. The first degree relatives are at a more risk, and the heritability was 32%. Means that at least in 32% is responsible for genetics is responsible. So in phobias, now a group at Columbia University had performed a series of analysis examining the specific phobia, social phobia, and agoraphobia, and their relationship with each other and panic disorder, and they reported higher rates of specific phobia in relatives of specific phobia probants compared with control, that is 31% to 9%. Higher rates of social phobia in relatives of social phobia probants, 16 versus 5%, and higher rates in agoraphobia relatives as well. And so now let us come to molecular genetics. So in molecular genetics, we'll be studying about linkage studies, genome-wide association studies, and candidate gene association studies. So linkage studies, as we all know, they are performed in family pedigrees, where with several affected individuals to identify the chromosomal loci which harbors a gene which influences a biologic trait or condition. Now, several linkage studies have been performed for anxiety disorders with few consistent findings, and suggestive evidences for linkage to PD has been reported for regions on chromosome 4q, 19q, 13q, 14q, and 22q. There are no published linkage scans for generalized anxiety disorder. Now, candidate gene association studies. Now, association studies allow one to test specific genetic markers for their contribution to a phenotype. That how a particular gene, which you select the prior knowledge, is contributes to a particular phenotype. To date, most association studies of anxiety disorders have focused on candidate genes, which have to be chosen using the prior knowledge, either from their position under a linkage gene or by their biologic relevance for the pathophysiology of the disease. Now, panic disorder has been the most frequently studied disorder compared to generalized anxiety disorder, social phobia, and specific phobia. The three most commonly studied genes are catechol O methyltransferase, serotonin transporter, and brain-derived neurotrophic factor. Now, let us come to genome-wide association studies. So, unlike candidate gene studies, which require a prior knowledge for gene selection, genome-wide association studies provide an unbiased survey of common genetic variation across an entire genome. So, genome-wide association studies, they require a very large sample of population for a good result. And so, the anxiety neurogenetic study was done, which had led to a large vast meta-analysis effort using data from nearly 20,000 subjects in 9 independent samples from the US, Europe, and Australia. The study has tentatively identified genome-wide association for 2 chromosomal regions containing potential novel anxiety disorders risk genes, a non-coding RNA locus, that is LOC152225 on 3q12.3, and the CAM-KMT gene on 2p21. Now let us come to gene environmental interaction. So gene into environment interaction occurs when the effect of genetic risk factors is modified by life experiences or other environmental exposure. For example, individuals with certain genetic risk profiles might be the most susceptible to anxiety-provoking effects of stress. So gene into environmental studies have mainly focused on serotonin, HPA axis, and the neurotrophin system in predicting ADs. The most extensively investigated gene is the serotonin transporter. Candidate genes in the HPA axis are also promising for the investigation of gene into environment, given the system's role in stress response, but it has been majorly studied for depression and PTSD with little focus on gene into environmental effects for other anxiety disorders. Now let us come to epigenetics. So epigenetics, we all know, it refers to potentially heritable and functionally relevant modifications of gene expression and chromatin structure accompanying no changes in genetic sequence. That is, whenever there is some environmental stress, there is changes in the genes, but the gene sequence remains same. But the changes are of methylation or acetylation of the histone proteins or DNA, and these are functionally relevant in the sense that they are heritable, but they are reversible. So this, I've already told that methylation, acetylation, phosphorylation, they are the molecular mechanisms of epigenetics. In this picture also, you can see that the pink one here represents acetylation. So when acetylation of the histone of the DNA, which is wrapped around histone protein, it occurs, it leads to condensation of the chromatin, and that leads to silencing of a gene. So animal models of anxiety disorder. So humans and rodents share notable similarities in their stress response and the brain circuits underlying threat detection and anxiety response. Therefore, anxiety disorders are especially well suited to be modeled in rodents. Cognitive capabilities are important in human stress response, especially in coping with stress. Therefore, while animal models of anxiety disorders aim to cover certain dimensions of the disorder, the full clinical symptomatology cannot be modeled in laboratory animals. Compared with human studies, a major advantage of animal models is the ability to tightly control the experimental studies, such as environmental exposures, and to have access to tissue samples at specific time points. So this I've already told in the previous slides, that what are the advantages and disadvantages of the rodent models versus humans. The environmental stressors used to induce anxiety-like behavior span from social to physical, from early life to adult stress, and are of varying durations. Despite the robust genetic contribution to anxiety in humans, most mouse models on stress and anxiety-like behavior are carried out with C57BL/6J and related strains, which are generally more resilient to stress compared with the other one, DBA/2J and BLB/CG inbred mouse strains, which are highly stress susceptible. Identification of genetic risk factors offers a route to develop etiologically valid animal models to understand the underlying neurobiological mechanisms and to test treatment responses. So these are my references. Thank you. Association with neurotropic factors. It has been seen. I've already mentioned about this. BDNF. So BDNF has been shown to seen when there is a substitution of methionine to valine. So the risk of anxiety disorders are increased in those areas, people who have this substitution. I did not find any study, but there may have. So thank you very much.